Thank you for the introduction, and thanks for coming out to my talk. So today I'll be presenting on my research that focuses on the effects of protein-coated microdietes on the growth and survival of larval California yellowtail, or Seriola dorsalis. Larval finfish culturalists face many obstacles in producing healthy young due to inadequate nutrition during early life stages. Growers experience high larval mortality and poor growth rates, in part due to their physiological limitations, which include developing digestive tracts, and developing sensory organs, as well as limited swimming ability. And additionally, their exact nutritional needs are also unknown. One available option for providing adequate nutrition to larval finfish, other than live feeds, are microparticle diets, pictured on the right. These are preferable in some aspects because they are consistent in quality, availability, and they're affordable and highly modifiable to the nutritional needs of certain species. However, these feeds do come with their own sets of issues. They are currently unable to fully replace live feeds, and they also break apart relatively quickly upon submersion, leading to rapid nutrient leaching. So the current microparticle diets are highly prone to nutrient leaching due to their high surface area to volume ratios caused by their small size and porosity. The images along the bottom show the resultant rapid nutrient leaching of sodium fluorescein, which we included into our house-made diets. And sodium fluorescein acts as a water-soluble nutrient proxy. And as you can see at the zero time point, there's very little nutrient leaching. And then by 2 minutes, there's already quite a bit. And then this is the 30 minutes. And so our solution is to protein coat the outside surfaces of these microdietes. On the left, you can see a matrix-bound microdiet that's uncoated, and then on the right, that's an encapsulated microdiet. However, there are some crucial aspects that need to be considered when designing these coatings. For instance, the particles must be protective enough to reduce leaching. However, they also must remain palatable and digestible to larval fish. For my project, I've broken down my research into 5 objectives. First, developing these protein coatings. Second, testing particle leaching. And third, particle stability, as well as conducting acceptability trials and growth studies to measure palatability and digestibility. So we'll begin with the protein coatings. Through benchtop trials, we formulated acceptable protein-based coatings. Then, using a fluidized bed coating machine, we coated off-the-shelf commercial microdietes by spraying on thin, even coatings shown in the diagram. As you can see, the spray nozzle here sprays up a mist using an atomizer of our formulated coating, and as the particles rise up with the air, they get coated, and then they umbrella out around to the outsides, and as they drop down, they dry, and then the process is repeated until we have our desired amount of coating on these feeds. Depicted here are some successfully coated microdietes and an uncoated microparticle diet under SEM. The furthest left images over here are the uncoated diet, and this is at 100 times magnification, and this photo is at 275 times magnification. And as you move right, we increase the coating thicknesses. Visually, you can see a reduction in surface jaggedness and porosity, so that's very promising. Next, we needed to formulate our lab-made open formula microdietes to be used for the remaining objectives of my research, alongside the commercial microdiet. The microdietes we produced ranged from 255 micron to 1.2 millimeters, and on the right is the formulation for our open formula diets. Next, we'll be moving on to particle leaching. So we ran both a taurine and sodium fluorescein leaching trial. We used taurine because it is a highly water-soluble, unbound-free amino acid shown to improve larval finfish growth. And we used sodium fluorescein because it acts as a suitable proxy for other water-soluble nutrients. In the bottom left images, you can see the leaching of sodium fluorescein of an uncoated microdiet compared to a coated microdiet at 5-minute time points. Both trials showed coated particles significantly reduced leaching, and in particular, the taurine leaching trial showed almost 2 times taurine retention compared to the uncoated diet, and at almost 4 times at the 5-minute time point. And then on to particle stability. To measure the particle stability, we submerged coated and uncoated diets in salt water and then ran them through the Mastersizer 3000 machine, which took measurements of particle size every minute. As you can see from the figure on the right, the blue coated diet curve showing the particle size over time was able to retain much more like larger particle size over time compared to the uncoated, which rapidly degraded and fell apart. Okay, next on to the larval acceptability. For the acceptability trial, we sent our feeds to Hub SeaWorld Research Center, Research Institute in San Diego, where we fed larval yellowtail for an hour, during which fish feeding behavior and acceptance of feed were recorded. We used both coated and uncoated commercial and open formula microdiets. And on the right is an image of the system that we were using. So through analysis of these results, we found that the coatings did not negatively impact the feeding incidence. We see increased feeding with inclusion of the feeding stimulants, butane and glycine. And we also saw that uncoated commercial diet, Otahime, showed the highest feeding incidence, but the difference wasn't statistically significant from the coated mem. However, there was a significant difference between the coated and uncoated Otahime, which would suggest that the effectiveness of these coatings may be matrix specific. And I just wanted to highlight the difference between the uncoated mem on the left and the coated mem, showing that the coated mem did do a little better than the uncoated. And lastly, we ran a growth trial at Hub SeaWorld. For the initial growth trial, we used 4 treatment groups, 1 coated and 2 equally formulated with hydrolyzed and intact casein as a protein replacement. And the last treatment diet we used was Otahime. These diets were administered to 5 tanks with, for a total of 20 tanks with 800 larvae in each. The larval Seriola were fed the microdiets from day 16 post-hatch to 52, and samples were taken throughout the trial. All right, so for the results of the growth study, we found that the coated diets performed slightly better than the other treatments, albeit not statistically significant. In the graph on the left, you can see age, days post-hatch on the x-axis, and individual dry weight on the y. And as you can see, the coated mem, which is the green, and the uncoated intact casein performed the best, but they were, again, not statistically significant. And on the right, you can see individual dry weight and standard length for each treatment at the final time point of the study, and you can see that the coated mem also did slightly better than the uncoated hydrolyzed mem. So overall, for my research thus far, we concluded that the coatings significantly improve particle stability and reduce nutrient leaching when compared to uncoated microdiets. The coatings are palatable and digestible to larval Seriola, and further research is required to optimize the surface coatings, which we plan to continue in future growth trials this year. We believe that this technology is commercially viable and scalable to other methods, or it's a scalable method for the industry, and it's applicable to a wide range of commercial type feeds. I'd like to thank my advisor, Dr. Matt Hawkyard, and my committee members, Gary Burr and Michael Hatesione, and as well as Hub SeaWorld and the Kingfish Company and the funding sources, National Sea Grant. as well as the University of Maine and the ARI. I'd like to thank Dr. Jim Hunt for help with feed analysis, or nutrient analysis, Kara Chuang for help both in just overall teaching me how to do basically everything, and Jordan Teska for assistance with technical support. Thank you.
Any questions?
Yes.
Okay.
So you started the feeding trial at 16 days post-hatch, correct?
Yes, that is correct.
Were they feed trained prior, or did you have to supplement with live feed to initiate feeding?
Yeah, so they were fed live feeds prior to day 16 rotifers and Artemia. And then we started to incorporate, or we fed them our microdiets along with live feed for a portion of the time.
Okay, and that was
Yeah.
after the 16 days, so they're still, like, during the growth trial, they're still getting some live feed?
For a brief period of the, yes.
We still have time for a couple of questions.
Yep.
You didn't mention what the coatings were. Can you quickly go through what
Yeah, so the
coating?
coatings that we decided specifically on for that, for our growth trial was a 5% by added weight protein coating, which included fish oil, the protein Zane, canola oil, as well as some feeding stimulants, which include glycine and butane.
It's not really just a coating.
Yeah, yeah. But protein-based coating is what, yeah.
Anybody else?
Yep.
So is that the recipe, is that in the publication? Or is the ratio of fat to protein?
It's not at the moment. It's still in the works, I believe. Yes. Yeah, it will be.
Good job. Thank you.
Yeah.
Yeah.
I was just curious if you noticed any behavioral changes now that there's more stable type of diet.
In terms of like fish behavior when they're being fed, so I actually wasn't able to be at the growth trials. I was only sending my feeds to Hub SeaWorld, but Kevin Stewart might be able to answer that. Yep.
So kind of add off that, did you notice any differences in water quality though too, since it, really doesn't really have.
Mm-hmm.
Anybody else? Okay, thank you.
All right. Thank you.